So darkly stained structure which you see in that nucleus, which is called as Barrbodies. Majority of the cases in Barrbodies lies in the periphery of the nucleus, and sometimes it can seen in other part. It can be seen in proximity of nucleolus. Majority of that cases Barrbodies presented in periphery nucleus. Then sometimes it can seen in other part. Proximity of nucleolus. It also referred to as X or sex chromatin. X chromatin or sex chromatin. So basically the Barrbodies deactivated or inactive X chromosome. So Barrbodies are deactivated or inactive X chromosome. This darkly stained structure named after Dr. Barr, who was a Canadian physician, who along with his student Dr. Bertram, Dr. Barr along with his student Bertram, 1948, identified this Barrbody structure. Now we need to understand as to why there is inactivation of the X chromosome. To understand this concept of why there is inactivation, let us see what is this Lyon's hypothesis. What is this Lyon hypothesis? This Lyon's hypothesis proposed by Dr. Lyon Mary, British genetics, in the year of 1925 to 2004. So according to this hypothesis, this hypothesis says female mammals, one of the two X chromosome in that somatic cell is inactive. So according to Lyon's hypothesis, female one of the two X chromosome in the somatic cell is body cell is inactive, and this inactivation either of X chromosome can be inactivated, and the inactivation of X chromosome is random, and this inactivation occurs during development, What we need to understand is that this inactive X chromosome remain inactivated in all the subsequent generation of this cell. This is Lyon hypothesis. So inactive X chromosome remain inactive in all the subsequent generation of this cell. Now why inactivation of X chromosome? There is inactivation to ensure female like males have one functional copy of X chromosome in each body cell. Let's understand. So basically this is to ensure female like males, they should have one functional copy of X chromosome in each body cell or somatic cell. This is to equalize dosage of gene product from X chromosome between XS female and XY male. Equalize dosage of gene product from X chromosome between XS female, XY male. So basically this X chromosome, this chromosome is inactive or disabled to avoid unwanted information from being passed down to future generation. So one X chromosome is disabled to avoid unwanted information from being passed down to future generation. Now there is X inactivation. Let's understand how this X chromosome inactivated. This is the process called transcriptional silencing. Transcriptional silencing. Let's understand that the X chromosome have a center called X inactivation center. X chromosome have a center name is called X inactivation center, XIC. They have two set of gene. What are the two set of gene? There are two set of gene. One of the gene inactive. One of the gene inactive specific transcript. XIST, inactive specific transcript. Okay. Another one is XIST reversed. This is the gene, and this inactivation is known as lionization. Inactivation name is called lionization. Normally, this active chromosome is enclosed within euchromatin. Active chromosome is enclosed within euchromatin, loosely packed chromatin. That euchromatin is loosely packed chromatin. And this euchromatin participate in the transcriptional activity. Now we know that given nucleus there are euchromatin, active chromatin and inactive chromatin. Active chromatin and inactive chromatin. Whereas inactive X chromosome is silenced by being packaged into that transcriptionally inactive structure which is referred to as heterochromatin. And this heterochromatin is tightly packed chromatin. We need to know that has no or little transcriptional activity. Now, what are the number of Barrbodies in the given nucleus? So, it is 1, less than 1. The number of chromosome, the total number of Barrbodies. The number of Barrbodies is 1 less than the total number X minus chromosome, nx minus 1, so X minus chromosome. For example, a normal male who has XY genotype, so the number of Barrbodies 1 minus 1, that is no Barrbodies, none. In Klinefelter syndrome, Klinefelter syndrome, you have 2X chromosome. So that number of bodies 2 minus 1 equal to 1. The normal female again, because we have 2. So chromosome 2 minus 1. So 1 being silenced. We have 1 Barrbodies. In Turner syndrome, where there is only 1 chromosome, so number of Barrbodies none. Then trisomy having 3 chromosomes, so 3 minus 1, 2. So total number of Barrbodies. So male, normal male none. Then this Klinefelter syndrome 1 Barrbodies, normal female having 1 Barrbodies. Then Turner syndrome none. Then trisomy having 2 Barrbodies. So now, how are these Barrbodies demonstrated? There are the best demonstrated. They are best demonstrated by any nuclear dyes. Nuclear dyes. They would be PAP or Greasel violet, then theanine, then orzine, then fusion, carbon fusing and fluorescent staining. Also used to study these Barrbodies, we can use phase contrast microscope. You can see Barrbodies even in unstained or in living cell. So, okay, we can see the Barr bodies in unstained or in living cells. So why Barr bodies testing is done? What are the indication for testing Barr bodies? One is the method to identify person gender, method to identify person gender, and to establish sexual identity in newborn and adult. So Barrbodies, why Barrbodies testing? Identify person gender, establish sexual identity in newborn and adult. Then this method was earlier used in competitive sports to identify male to identified male masquerading as female to give competitive advantages. Though this particular test is not being done, routinely, it was a method to identify a person gender. As screening test, genitalia, sexual character are not clear. So then forensic in crime scene, you know where you can find that evidence of our bodies in buccal epithelial cell in trace of saliva. Sometimes Barrbodies testing is asked whether there is a delayed onset of puberty and a crime scene. So why bar body testing is done? Sexual identity in newborn, general identification, then delayed onset of puberty and a crime scene. Next, what are the procedure is taken a sample. The most common smear which are used to known study bar bodies are this buccal smear. You ask the patient to rinse the mouth, clean that mouth, and then take a smear from the buccal mucosa either using wooden spatula or even a cyto brush, and then smear is made on that glass slide. We can to fix using fixative and then staining using those staining, and we can observe under microscope. So we can observe squamous cell in the buccal cavity. We can identify that presence of Barr bodies as a darkly stained structure in that periphery of that nucleus. So these Barr bodies are seen in 30 to 60 percentage of female stomatic cells. What are the other tissue where which from Barr bodies can be studied? They can be studied from corneal epithelium, vaginal mucosa, fibroblast. Other tissue from which Barr bodies can be studied in corneal epithelium, vaginal mucosa, and fibroblast. This Barr body can also be studied in polymarbonuclear leukocytes, mainly neutrophil in peripheral blood smear. These Barr bodies are seen in variable form, mainly drumstick appearance. This one is the drumstick appearance. The drumstick are small, deeply stained nuclear mass about 1.5 micron in diameter, attached to that body of that nucleus by means a thin stalk. It is seen in 1 to 17 percentage neutrophil with an average of 2.9 percentage. The drumstick first identified by Professor William Markey Davidson in the year of 1950. He was that Scotland pathologist and histologist. So it also called as Davidson body. Drumstick appearance is also called Davidson body. So that the reason why this one also refer to Davidson body. Barr bodies seen in important things we want to remember here. Barr bodies seen in buccal smear. Davidson body seen in peripheral smear. Then Other form of Barrbodies in peripheral smear, which can be in that form of rocket. It can be seen in that form of these sessile nodules, small club and minor lobe. Most commonly, but most common form is the drumstick appearance only. So, summary or different between these bar bodies or Davidson bodies. Bar bodies found in stomatic cell in females. Davidson body presented in polymarbonuclear leukocytes in female. Then bar bodies seen in buccal smear. Then Davidson body seen in peripheral smear. In this bar bodies in squamous cell epithelium, this one neutrophil. So these are the things in Barr bodies and Davidson bodies. Thanks for watching.